So yes, today I'll be talking about the use of solubility maps in the development of robust crystallizations. The presentation will focus on ways of measuring and regressing solubility in multi-component systems. Also, at the end of the webinar, I'll discuss some of the practical aspects of using DynoChem to develop a solubility model in a multi-component mixture. Reproducibility upon scale-up at the center point conditions. So we ran 3 batches at 70 kilos each along these center point conditions and monitored by FTIR. And what we see is very consistent batch concentration across the 3 batches throughout each of the unit operations of this crystallization. We were also able to monitor relative supersaturation, and that was in line with what we'd expect based on the lab data, and it was well controlled at less than 50%. So this gave us confidence that the process is reproducible and well controlled. And the next slide, this slide shows some of the product attributes that are measured that are critical for drug product processing. So particle size distribution is very consistent across the 3 batches. The bulk density was consistent and in the range that was desired and in line with lab expectations. So it showed that we have good confidence in this process, and solubility was a key contributor to that confidence. So in conclusion, for the first part, knowledge of solubility map is vital for defining robust parameter ranges and identifying process risks. Automation equipment was quite useful here, and coupled with the use of an internal standard, we were able to obtain extensive and accurate weight % solubility measurements from this complex system in a fairly short period of time. DynoChem was a quite valuable tool to regress the solubility, which is in the fitting module, and then defining the seeding and crystallization window in the multivariate space, and that's the simulation portion. So from DynoChem, we can rapidly screen nonlinear solubility models in various parameters, and then directly incorporate the solubility model into crystallization simulations. So that concludes the first part of my presentation, and then the second is some of the practical aspects of how to use DynoChem for solubility regression. And I'll go into that now. So the first part will be discussing the current solubility model that exists in DynoChem, followed by a suggested strategy for fitting solubility data, especially when there are a number of potential parameters that you want to fit. And then a couple examples of using the solubility data to evaluate the crystallization. Another item to look at is the correlation matrix, and you can see that in cases where the correlation is close to 1 or -1, that could be a flag for overfitting. In this case, which is the case with this D-methanol term, where it's strongly correlated with the main term for methanol. We kept it in the model since it did help with the model fit, but I think that is a point for discussion. So a couple of tips, like I mentioned, reducing the model to the necessary number of factors is key in order to avoid overfitting, and you want to remove terms with high confidence intervals. Another thing that I haven't mentioned yet, but it's important, you'll notice that heptane is not in this series of parameters, and that's because heptane is the nth component. So we want to include at most n-1 components, because the system is fully defined with n-1 components specified, because the nth component can be defined as a sum of the other components present. So if you do include, say, the nth component in your fit, then your entire model will be overfit for 1 and will have very high confidence intervals. And finally, how can we use these solubility models? So I touched on that in the first part of the presentation. This is a more detailed view of that. So in this case, we can look at how the solubility changes as a function of the crystallization progress. So in red, you can see how temperature changes during the crystallization, and then in yellow, you can see how heptane volume percent changes during the crystallization. And then the effects of solubility from those changes can be seen here. So this is just sort of a high-level view of how solubility changes during the crystallization, which can be useful in itself. Then one final slide here is something that can be put together pretty quickly from the solubility model, which is this is the chart that you saw on the previous slide. Then we can see how yield changes, so sort of instant yield or generation of product crystals as the crystallization progresses. That can be useful. And then one that is quite useful, sort of as a very gross approximation, is the rate of mass deposition in terms of percent of total per minute. This assumes basically instantaneous crystallization of your product, so there's no supersaturation that's generated. Crystals are formed immediately once the solution becomes supersaturated. But this can be a useful sort of first glance at finding potential risk points for a crystallization, where you'd want to have, if possible, have as smooth of a rate of mass deposition as possible. And if you do see issues in a crystallization, spikes in the rate of mass deposition may be a place to look first as a potential pinch point.